me. Okay, so the plasma membrane, it is also known as cytoplasmic membrane, is made out of a phospholipid bilayer, and then it has embedded in this bilayer proteins. They can be either peripheral, found at the internal or external leaflet of this phospholipid bilayer, or it can be integral or transmembrane proteins. Basically, this will be spanning across the two leaflets of the membrane. And then some of these transmembrane proteins will have a gap in the middle, which act as a channel. So through this channel, molecules can come in and out. Now, transmembrane proteins, they have a different function, including adhering the cells to enzymes that they might have internally, or adherent enzymes that the cells have internally to these transmembrane proteins, or they can serve as proteins that can bind ligand, so that the cells can receive a signal from the environment, and the cells can make sense of this environment. Now, sometimes these proteins can be combined with other molecules, like a lipid. In this case, you would call it if the lipids are combined with a carbohydrate, in this case, you will have a glycolipid. And sometimes these proteins can be combined with a carbohydrate, and this, in this case, will be a glycoprotein. An example of these glycolipids can be this lipid A that we saw before, that is part of the gram-negatives. And then here is a figure showing you this plasma membrane in a transmission electron micrograph. In this case, we have this plasma membrane shown in blue color, and then you see the peptidoglycan in red color, and then you see a second membrane outside of this peptidoglycan. So, a bacteria that has two membranes, it is classified as gram-negative. And it has the sandwich in between these two membranes, which is the peptidoglycan. And the space between these two membranes is called the periplasmic space. So this is an image then of a gram-negative microorganism, Vibrio cholera to be more specific. And then here is a cartoon showing you this plasma membrane with its component. So this is the phospholipid bilayer, the external and the internal leaflets. And then, of course, we know that these phospholipids have two fatty acid tails that faces internally because these are hydrophobic. And the phosphate heads, which has a negative charge, can combine with water or can interact with water. So these will be charged molecules. So these are hydrophilic, while these fatty acids are hydrophobic. And then you see here some transmembrane proteins, like in this case you have one, another one, and this channel. And then again, sometimes these transmembrane proteins can attach internally or externally some proteins, including enzymes, that can help for the microorganisms to perform any function that they're supposed to perform. And here is a magnification of these phospholipids, again, the fatty acid tails, two specifically with this phosphate group. I don't know if they might show in this animation the nature of these phospholipids, which is moving, and that's the fluid mosaic layer. So let's pretend that I'm a phospholipid, and then my lower limbs will be the fatty acid tails, and I'm the polar head. So I will be doing this, rotating, and also I will be moving to the side. So that's a fluid mosaic model. Proteins as well, they are not static. So let me see if this one might have something about this. Well, it shows partially uh, transport, but it didn't show how much movement you have within this plasma membrane, which is okay. So, uh, so this membrane, because of this lipid nature, is fluid, it's flimsy in a way, and part of these proteins, it helps for this plasma membrane to have rigidity, so it won't be super flimsy. And then these phospholipids, again, rotate and move laterally and can be self-sealing. So, because we have this phospholipid bilayer in these channels, then you expect that you have a compartment. Not everything can come in and out of the cells or into the cells. So then you as well have in this plasma membrane enzymes in the case of these microorganisms that helps to produce ATP. So remember, eukaryotic cells, they have the mitochondria, where we have in the matrix the enzymes either in the Krebs cycle, and then we have the electron transport chain that helps to produce ATP. But microorganisms, they don't have mitochondria. So then they have these enzymes embedded in this plasma membrane. Some of these membranes will have foldings, and inside these foldings, they have pigments, photosynthetic pigments, that makes this bacteria to have a particular color. And these will be the chromatophores. I'm not going to play that. So here we have some of these structures like these chromatophores. And this is like a view internally. So what you can think of is sometimes these pigments also can be internalized, or they can remain within the plasma membrane. No matter what it is, if you look them internally, it is either because they were pinched off from this plasma membrane, or because when they took the TEM, you're looking the bottom part of the plasma membrane with these protrusions. It's a two-dimensional view. So if you see the lateral view, maybe you will see those foldings. Now, because we know that these lipids can be damaged with chemical agents, we actually can use them to control the growth of microorganisms. So one of these chemical agents that we can use is alcohol. And alcohol, basically what it does is dissolve the phospholipids. And that creates pores that makes the cytoplasm to leak, and then microorganisms cannot grow without having a complete plasma membrane. Another one would be a detergent, positively charged, like quaternary ammonium or quat. So this one, it's a very good disinfectant as well that can be used to control microbial growth, and some antibiotics like polymyxin. This polymyxin, it is found sometimes, not always, but sometimes, it is found in combination with two other agents in what we call the triple antibiotic ointment. It's a topical drug. And then what it does, just like similarly to alcohol, it creates pores within the plasma membrane, and then the microorganisms won't grow. Okay, so passive processes and active processes in the cells. The difference is that in passive process, you don't use energy. That's why it's passive. And then, since you don't use energy, the molecules will move in the direction that favors the movement, basically from more concentration to low concentration. We are right now 8 people in this room, and then 3 are here on this side of the table, and 5 are over that side of the table, or those tables that you have there. If we were about to move in passive process, it will be from more to less, from 5 to 3, until you reach equilibrium. So how many people will have to move to make it even, and then we don't move anymore? Only 1. So that's passive process. Active process is against concentration gradient. So you spend energy. And here it's hard to compare right now, but it is something like if someone is going to go into the elevator and the elevator is packed already, but the person definitely needs to be in its appointment in the 20th floor in a big city. And then the person sees that the elevator door opens, and then he has to press himself against the crowd, although the crowd will be like all grouchy. But you have to do that. You have to spend energy to go against. We won't practice that now. It's like if we as 3, we were against all of you, and then you would be like, no, you can't. So that's active process. And then simple diffusion. Basically, molecules that are how can I put it? So molecules like oxygen and CO2, ethanol, can move across the plasma membrane because they are not hydrophobic. So they can cross through this phospholipid bilayer without a problem. And here is the typical figure showing you what happens with time when you introduce into a beaker with water a crystal salt with color or dye in it. So you put it in the bottom of that beaker, and then at the beginning, of course, since you have more concentration in here, oops, so the dye will start moving from bottom to top until you have, with time, all the water colored in the same way. So that's simple diffusion. And then here we have molecules moving in and out, like oxygen, CO2, ethanol. You don't need a channel in that case, and or transporter. Now, everything is kind of related as well to charges, size, and shape. So, water will have specific channels to move across the membrane. So these channels are called aquaporins. Aqua for water in Latin, which is aqua. And then water will be following specific gradients of solutes. Like water will move from a low concentration of solutes to a high concentration of solutes to try to dilute these solutes if it's needed. Specifically, more will be like glucose or sodium itself. And then if a molecule has a charge, either positive or negative, the channels internally, if it's that one that moves across the channels, it will have to have a specific amino acids with a specific charge that allows this molecule to move. But let's say that you have the right charge, and if it will be, for instance, an anion, which is a negative charge, let's say that you have the channel that allows the passage of this anion, that it can have maybe positive charges, kind of not so much combined with negative charges amino acids, because otherwise the ion would get stuck in the middle of the channel. But then you have to have also, if you have another ion, you have to have a specific shape or size. Not everything that is negatively charged can pass through there. So there are several factors that plays a role. And then in some cases, you will have what is called facilitated diffusion. So some channels, they have a gap in between that holds temporarily this molecule to help it to carry through this channel to go into the other side. Example would be the glucose transporter. When you grab a glucose molecule inside the channel, you close the outside of the channel, and then you open the inside, you pass it, and then you close it again. And some of these channels will have little doors or gates that open and close, allowing the passage of molecules, not freely, but whenever it's needed. So some transporters are non-specific and others are more specific. So facilitated diffusion, it is carrying a molecule. There still are gentlemen. But back in the day when there was a pond of water, Maybe that gentleman carry the lady and then put it on the other side of the pond. Nowadays, both of them, they go to the pond, or maybe the lady piggyback the guy. I don't know. So that's facilitated diffusion, carrying someone. Okay. Move it across. So here we have examples of non-specific transporter versus a specific transporter. Non-specific doesn't necessarily mean that anything can move across. In this case, it's a cation channel. So positively charged molecules can cross, and then again, it has to be the right size. Not everything can move across. So it will be, for instance, in this case, let's say a sodium channel instead of a potassium channel. Your gentleman can only move across you, grandma, mom, someone that you know, not every lady that moves across. I don't know about you, but I wouldn't let it go like that. Want to get into trouble, being unspecific. And then in here we have the glucose transporter. Again, we have this little gap to catch this big molecule, glucose. And then this area is going to seal, and then it opens here, this part. It takes a while for this facilitated diffusion process to happen, but also that leaves time as well for metabolic processes inside the cell to take place, like utilization of this glucose. And osmosis, again, moves from an area of higher water concentration, which means that you have lower solute level, to an area of low water concentration where you have more higher levels of solute. And my typical illustration about this is you wake up in the morning, you're serving your cup of coffee, you previously added your tablespoon of sugar, and then you pour your coffee, your cream, and then you ask yourself, because you were half awake, did I add sugar already? And then, well, you add another tablespoon. You move it, you swirl it, and then you taste it. It tastes sweeter than what you like it, usually. It's too sweet. So what would you do? Because he has high concentration of a solute, in this case glucose, well, you will pour more coffee. So you will dilute it. So that's the purpose of osmosis. Yeah, so we have these aquaporins. The levels of aquaporins will vary. You will transcribe, make more channels when you need to have this process going, and then you shut them down. You make no more aquaporins, and you can recycle them. And since water is not going to be moving always without limit, you have to stop movement of water with what we call osmotic pressure. So here we have the example of osmotic pressure. So you have a beaker with water up to this level, almost full, and then you introduce a capillary vessel, a glass capillary, with your cellophane bag wrapped around, and inside this cellophane bag, you have a solution with solutes. In In this case, it is high concentration solid. Let's say it's 5% glucose. And then outside, you have only water. And this membrane doesn't allow the glucose, these big molecules, to go out. But it's semi-permeable. It has gaps in its layer to allow the movement of water in and out. So you introduce your fill bag with 5% glucose solution, and then you let it stand with time. So with time, water from the beaker will go into this cellophane bag, it will expand, and then your water column will rise up to this level. When you reach equal concentration of water here and here, that will be the equilibrium. So don't think that the water will move indefinitely. So you will be having the water coming out of this tube. So then you will reach equilibrium. So that's osmotic pressure, preventing more water to move out. So microorganisms, just as our cells in our body, or in an animal, of course, any cell, when it's alive, may encounter 3 different types of environments. An environment where you have an isotonic solution in which that solution can be equal in terms of solids and water concentration to the inside of the cells. And in this case, in this isotonic solution, you will have movement of water in and out of the cells, of course, because you have to transport other molecules like hormones, nutrients, but the cell will remain at the same size because it will not gain volume. And well, the same size, when I'm talking about the same size, is not gaining water or losing water. Of course, if it's a cell that will grow like muscle cells, if someone is doing exercise, having more growth hormone, yeah, that cell is going to increase in size. But not related to having excess of water coming in. Now, another environment that a cell can encounter, it is actually being surrounded in a hypotonic solution. Hypo means low. So in this case, you have low concentration of a solute. And then because you have more water outside the cells than in, then the water moves from outside of the cells to into the cells in the cytoplasm to dilute this high concentration of solutes inside the cell. So the water will move into the cells and the cells will swell. If it's too much, eventually the cells will burst, and this is called osmotic lysis. Now, in a hypertonic solution, you have the opposite. You have a lot of concentration of solutes outside of the cells, and then your cells will donate water, will move the water from the cytoplasm into the extracellular environment so that you can dilute this excess of solutes. And as well, my typical illustration is someone who goes to the movies, get popcorn, and get its soda, sugar drink, that is not diet, of course. And then what in the movies they do? They give you free popcorn, but they don't give you free sodas. Guess what? Well, because soda is very cheap for them, and then they sell it expensive to you, and most of the time, at least the container, they give it to you like half filled with ice. So you're paying double for whatever you're getting. But then you start eating your popcorn, you are hungry, and then you feel thirsty. You drink soda. Well, you're adding sodium from the popcorn, and then a lot of sugar from the soda. So you start feeling more thirsty and you keep buying and buying. And that's good business. And then you go out, eat pizza, and maybe you had a ice cream cone. You go home, super thirsty, start drinking water. That's when your thirst stops. Because you are diluting the excess of hypertonic solution that you have in you. So, this is the principle of osmosis. Yeah, in isotonic solution, water come in and out, but the cell has the same volume. Iso means same. Hypotonic solution, basically these little dots represents the solute, so not so much solutes, a lot of water, all this blue color. So our cells will gain volume and eventually will burst, osmotic lysis. And then for microorganisms specifically, when you put them in a hypertonic solution, you will have a lot of solutes, as you can see there, those bunch of dots. And then the cell will start losing water. So it will donate water to the extracellular environment, excuse me, and you will have detachment of the plasma membrane, and that will lead to cell death. So this is called plasmolysis. There are certain characteristics of these contact between the plasma membrane and the cell wall that maintains the cell alive. When people didn't have fridges back then, how did they preserve the food? By adding a lot of salt or a lot of sugar. Okay, so they created this hypertonic environment and bacteria didn't grow. So those were passive mode of transport. Now let's talk about active transport. So again, we require energy, and that energy will be in the form of ATP. You will be moving substances against their concentration gradient from lower to higher. And then you have something that is called a mode of transport that is called group translocation. So in this case, you require a transporter protein and phosphoenol pyruvic acid. And then as you move this molecule, the molecule will be altered. See, I want to see that. So we have in active transport molecules that only transport one molecule across in one direction, some that moves molecules in the opposite direction. These are called antiports. And then symports will be molecules that are moved basically simultaneously. So let's see what this animation says. So, let's see. Which agents can damage the plasma membrane in bacteria? Mm-hmm. alcohol in detergents. How are simple and facilitate diffusion similar?
They don't use ATP.
How they are different?
Facilitated uses proteins and channels to help move stuff across the membrane.
And you have physical contact, right? In simple diffusion, you don't have physical contact with these channels. Okay, so that was the plasma membrane. Now the cytoplasm. So the cytoplasm, it is this medium that has water. It might have some inclusions and structures that allows the cells to function, like ribosomes, DNA, and cytoskeletal proteins. Cytoskeletal proteins helps for direct molecules across, maybe will help in movement, like for instance the pseudopods from protozoans. They basically crawl because they have extensions of the cytoplasm to help them to move. And then some of these can be helpful for moving the DNA, for growth. Of course, we know that it's important for maintaining cell shape, especially in eukaryotic cells. In prokaryotic cells, the rigidity of this cell wall is what it helps to maintain the shape. And also aids in cell division because part of these cytoskeletal proteins, at least in eukaryotic cells, of course not in bacteria, with the microtubules, which is part of the cytoskeleton, you grab the middle of this duplicated DNA, the chromosomes, the kinetochore, and then you pull them apart so that you can give equal number of chromosomes to each daughter cell during cell division. So basically you form the mitotic spindle. So now the composition of the cytoplasm will vary depending on the microorganism. Some microorganisms will have some inclusions in there that helps them to store lipids. Other ones, they like to store sulfur, etc. So, and then also concentration of ions vary not only between the different species of bacteria, but depending on whether the bacteria already was basically found in an environment rich in nutrients or it acquired nutrients. So basically, these other things, which is not water, not cytoskeleton, it is not related to nutrient content inside this cytoplasm. Now, prokaryotes, they don't have a nuclear membrane, and there is one exception. We will see that in a couple of chapters. But basically, most of them, they don't have a DNA enclosed by a nuclear membrane. But the space in the cytoplasm where this DNA is located is called the nucleus. Can it be static? Like, can we have the nucleus found only in one end, always, in the cytoplasm? Believe me, well, you will believe me, I don't know. But that's something interesting. Why? Because maybe, yeah, that nucleus moves to one side or the other. There might be signals that helps that movement to happen. And that would be something interesting that someone can study with DIC or differential interfering contrast or phase contrast, basically microscopy. For sure, in protozoans, they study the movement of vacuoles. And that one, it's very easy microorganism to study. So I'm pretty sure that that's a eukaryotic cell. They will have movement of their nucleus. So remember that the bacterial chromosome is typically one single chromosome, circular, double-stranded for sure, and we don't have wrapping around of this DNA around histone proteins, just like as in eukaryotic cells. And not all the microorganisms, but some, they will have an extra piece of chromosome or extra chromosomal DNA that contains not so many genes, between 5 to 100, but these genes are super important for their survival. Bacteria that has plasmid genes can survive more aggressions. So they might have genes that prevents them from being destroyed by antibiotic disinfectants, or maybe they are able to produce a toxin. And we will see how these genes are transferred from bacteria to bacteria. It's called horizontal transfer, but we will see as well how these genes are coming from sometimes from being infected with a phage, which is a virus that infects bacteria. Every single cell, no matter which cell it has to have a protein factory, so it has to have ribosomes. Bacteria, prokaryotes are not the exception. So bacterial cells have a lot of ribosomes. And these ribosomes, just as any other ribosome, are made out of a protein and a ribosomal RNA. It will be interesting to know as well where they are made, because bacteria, they don't have nucleolus like our eukaryotic cells. Where does the ribosomes coming from? And one thing for sure is that these ribosomes are 70S. So eukaryotic cells and eukaryotic cells have free ribosomes or attached ribosomes to the rough endoplasmic reticulum that are 80S. And then these 70S have a couple of subunits, one that is large, that is 50S, and another one that is small, that is 30S. So both of them, they form a complex, and then they are called the 70S. And no, in here is not arithmetic problem, like maybe why 50 plus 30 doesn't make 80S, no. S stands for Sedberg units. So it is a unit that tells you how likely the material that you are centrifuging, ultracentrifuging, goes to the bottom. So heavy objects go first to the bottom, and lighter objects will stay on top. So let's say that we are going to be centrifuging some ribosomes from bacteria. So then what you expect to have in this layer is the 50S subunit, and in here will be 30S subunit. And then as a whole, if you didn't separate the subunits, they will stay uniformly against maybe other molecules from the from the cytoplasm, and that makes it an 70S. So it's a unit of centrifugation, basically. And now, one target for preventing microbial growth, because ribosomes are very important for bacteria, it is to basically prevent these ribosomes to function. And you'll have several antibiotics that interferes with the function of these ribosomes, including streptomycin, gentamicin, erythromycin, and chloramphenicol. That's the last chapter that we're going to talk about antibiotics. So we will see the mode of action, but basically these 4 antibiotics are disruptors of the synthesis of proteins. And here is the illustration showing you these ribosomes and its subunits. So 30S, smaller, 50S, larger, stay on top of each other. They form like a little gap in between. That's basically the gap that allows or like a little tunnel where you have messenger RNA running so that you can read this messenger RNA and then you can start forming your proteins by adding the proper amino acids. So in the cytoplasm, bacteria not only have then the nucleoid and the ribosome, but also have inclusions. And these inclusions will be named differently depending on what they are, and most of the times they carry the name of the type of molecule that they have inside. Some of them, they will have phosphate reserves, and these are called volutin or metachromatic granules, depending on the combination of phosphate with other molecules. These granules change in color. That's why they're called metachromatic. And then some of them, they will have granules of polysaccharides that they use as a source of energy. Phosphate groups, the bacteria can get from phospholipid membranes from the cells that they might be infecting, or from nutrients that they have in the environment. Polysaccharide granules, similarly, from the environment, they can grab these saccharides and they store them. It's like the glycogen for animal cells or the starch for plant cells. Lipid inclusions, sulfur granules, both of them are energy reserves. So in a way, depending on the environment where this bacteria is living, it is what type of inclusions it's going to have that tells you that these are adaptable to the environment so that they can survive. Carboxysome, they have this enzyme, rubisco, that it is important for fixation of CO2 during photosynthesis. Some of them are found in liquid environments like lakes or oceans, and they use little cylinders, protein cylinders, that helps to trap gas. And that gas they can compress or decompress, so you can have more pressure, less pressure, and that helps them to move closer to the surface of the water or away, so that if it's a photosynthetic bacteria, during daytime, they go to the surface, they can produce photosynthesis, or they undergo the process of photosynthesis, and then during nighttime, they can go down into the bottom of the ocean or the lake. So they have buoyancy, basically the ability to move at different levels in an aquatic environment. Some of them, they can store forms of iron, like iron oxide inclusions, and these are called magnetosomes. And these magnetosomes can be used to destroy toxic form of oxygen, like hydrogen peroxide or H2O2. And it's called magnetosome because if you put some cultures of bacteria and you have in the bottom a magnet, you can clump them there. Something interesting, of course, is because iron, it can give you certain color. I mean, that's why it gives our blood the color when it's combined with oxygen or CO2, specific brightness of red. But look at this bacteria. This is a rod with Amphitrachus flagella, so it's two flagella on two endings. But look here in the middle. This is a transmission electron micrograph. These vacuoles, these inclusions, they have iron oxide, so it changed red, and that's the color of iron oxide. Now for endospores. So endospores are basically the form of survival for bacteria. Sometimes bacteria are just plainly run out of nutrients, and it senses in the environment the lack of nutrients. It starts duplicating its DNA, and then it will encase it in a double membrane, and of course the cell wall, and calcium salts, and they will be released. So these forms of bacteria will be resistant to desiccation, basically losing water, dehydration, high heat, chemicals, and radiation. Once they encounter the right environment, they can go back. So it's not a reproductive process, it's a survival mechanism. Two species of bacteria can produce endospores. Bacillus species or genera, Bacillus and Clostridium. Why is this? Well, they're rods. They have the right shape, and they also have much volume than a spherical bacteria, like coccus. Coccus don't produce endospores. And then the process of actually making the endospores is called sporulation, and germination is basically regrowing the bacteria after it finds the right environment. So the cans of food that we consume, they are processed specifically to prevent having contamination with spores. So they put the food, they process it with a specific machines, and then they seal it. If you don't have spores contaminating these cans, and some of these species, specifically more Clostridium, it's a gas-producing bacteria. If your canned good is not contaminated with spores of Clostridia, you don't expect to have this endospore growing into a vegetative form, and then you don't expect formation of gas. So your can will always be the right size, the right shape. But if you see a can that has perhaps the lid like bulging or a part of it, I mean, unless you're desperate, you can eat it, but I wouldn't. And yes, sometimes they can fall. I don't know, that thing may make the air that sometimes may have inside to be bulging. You don't know. I mean, I would say do not run the risk. And then here we have a TM of Bacillus subtilis. It's a rod. So this bacteria, this is the bacteria, this is the cytoplasm, has internally this endospore. So this is the formation then of the endospores, the sporulation process. First step will be to duplicate your DNA, and in this case, see how the nucleoid, of course, can move around. It goes to one end. And then you will start forming a wall made of phospholipids. Why? Because you want to wrap around your DNA in here. So that's the first step. And then the second step is completely wrapping around this DNA. And then you, in the third step, you add a double wrapping. You add another plasma membrane. In the fourth step, you form that peptidoglycan layer between these two plasma membranes. Last step will be release. So you form a coat, and then that coat is going to basically wrap around this endospore, and then you will have a free spore. And of course, mother cell has destruction of its DNA. Something that I was thinking about now, it is the process of sporulation. Kind of, if you were about to see it this way, like you have your first plasma membrane, right? You have your second plasma membrane, and then you add a peptidoglycan layer in between. Only gram-positives, like Bacillus and Clostridia, or gram-positive bacteria, produces these endospores. But if you see this type of wrapping of the two plasma membranes and the peptidoglycan, it reminds you about a gram-negative, because these are the only ones that has two double membranes and then the peptidoglycan. One way to distinguish, well, first the endospores, so the spores are smaller than actually a bacterial cell itself. And this coat that you have outside that protects this cell, or well, this spore, it has also other molecules that helps it to distinguish from, let's say, an encapsulated gram-negative, because it kind of will look like an encapsulated gram-negative. So you have molecules like calcium salt, the picolinic acid that are specific for these endospores. And then maybe if you measure the thickness of the peptidoglycan, it might be more than 3 layers of peptidoglycan, which basically distinguish it from a gram-negative. Okay, so let's see our review questions. Where is the DNA located in the prokaryotic cell? What is the name of that structure or that place where you hold it? Nucleoid. Mm-hmm. What is the general function of inclusions?
Storage of excess nutrients.
Mm-hmm. And then under what conditions do spores form?
Nutrient depletion.
Mm-hmm. Nutrient depletion. Actually, not only that, but I don't know exactly how I said it before, but if the bacteria also run out of water, if the bacteria is subject to harsh environments like excessive heat or radiation, you can form these endospores. Okay, now let's see characteristics of eukaryotic cells. So in here we have an image showing you major structures of eukaryotes, including plant cells here from this part, and then animal cells from this part. Any eukaryotic cell has a wrapped nuclear, nuclear material. So you have a nucleus, and then you have a nucleolus, and you have next to this the rough endoplasmic reticulum that basically receives part of the messenger RNA that comes from the nucleus after transcription so that you can make proteins. Of course, not all the proteins are made in the rough endoplasmic reticulum. You have free ribosomes. So that messenger RNA goes there, it will be made into proteins. Now, we also have this other organelle that similarly as to the rough endoplasmic reticulum, it is made out of a sac of membranes. And this is the smooth endoplasmic reticulum that majorly helps to detoxify the cells and also makes phospholipid bilayer in the case of eukaryotic cells. And then you have two centrioles. These centrioles act as an organizing center for the microtubules when the cell is about to divide in mitosis. So they migrate into different poles, and then they attach the microtubules that are going to attach into the kinetochores of the chromosomes, and then they are pulled apart. We have the UPS from the cell, which is the Golgi apparatus. Golgi apparatus receives the proteins from the rough endoplasmic reticulum. So the rough endoplasmic reticulum releases vacuoles that they are now merged into the sac of membranes as well, which is the Golgi. So what three organelles have a sac of membranes will be Golgi, rough endoplasmic reticulum, and smooth endoplasmic reticulum. So then the rough endoplasmic reticulum basically makes the proteins, put them into a vacuole, and then transport them into the UPS, into the Golgi apparatus. The Golgi apparatus put a tag, maybe glycosylate the proteins. And then with this tag, these proteins go into specific parts of the cell. It can be going into a channel, or maybe it can be exported. So this is the distribution center then. And then this Golgi also makes some inclusions that holds enzymes, the lysosomes. And then of course we have the powerhouse of the eukaryotic cells, which is the mitochondria. Mitochondria is very unique because it has its own DNA, it has its own proteins, and the ribosomes that it has, it's 70S. While the ribosomes that eukaryotic cells have is 80S. So that's when they came with this theory of endosymbiosis, apparently saying that mitochondria previously was a bacteria that was engulfed by the cells, and then they become the mitochondria. And we also have, of course, cytoskeletal proteins that makes this cell to have a specific shape. Of course, we have a plasma membrane, and then we have sometimes in some cells extensions like flagella. In animal cells, human cells, the only cells that have flagella will be the sperm cells. And sometimes we can have cilia, like in the case of our throat cells. We can have pili, we can have microvilli that helps to absorb nutrients. And then plant cells, they have large vacuoles, they have mitochondria, they have chloroplast, which is an organelle that it is rich in chlorophyll inside the thylakoids, which are similar to the cristae in the mitochondria where you produce photosynthesis. So here is the transmission electron micrograph of a plant cell and an animal cell. So you have nucleus, nucleolus, chloroplast here, and then you have mitochondria. And then large vacuoles. In the case of DNA, of this image, it's very magnified only towards here to the nucleus, but we have this nucleolus, and then you have the nucleoplasm surrounded by this nuclear membrane, you have nuclear pores, and then you have your chromatin or your DNA in there. And this is a mitochondria, rough in the plasmic, in the plasmic reticulum. It doesn't say specifically which one can be. I'm pretty sure that this has to be the smooth, just because it's not as close to the nucleus than the rough, which I'm pretty sure is this part. And then somewhere there, of course, is the other organelles. So I think we're running out of time. Well, we have 2 minutes. So now let's look at the differences between prokaryotes and eukaryotes. So bacteria can have flagella just as animal cells, so these are similarities. And they are few in number, while cilia are short projections and they're numerous. Now, bacteria don't have cilia. Animal cells have cilia. So functions of both of them for locomotion. That zigzag motion of the flagella helps for the sperm cells to migrate. And then cilia helps to move substances across the plasma membrane. Across the cells, like the mucus that moves in our throat. Here is an example of cilia and flagella. So this is a long extension flagella, cilia in this paramecium. It's a eukaryotic cell. It has a lot of cilia. It is also used as a form of locomotion for them. And that would be all.